This presentation is for informational purposes only and does not constitute medical advice. If you are considering prescription medications such as Ozempic or Wegovy, consult a licensed healthcare professional before making any decisions. There has been an intense surge of public interest in GLP-1 based drugs for weight loss and metabolic health. At the same time, many people wonder whether it is possible to leverage the body's own GLP-1 system without pharmacology. More recently, researchers have identified a naturally occurring peptide that appears to regulate appetite through a different pathway. The practical and scientific implications are important. If appetite and glucose control can be influenced more precisely, interventions might be more effective and better tolerated. This video walks through what the research shows, what GLP-1 actually does, how your biology generates it, and realistic ways to amplify those natural signals. It will also examine the BRP discovery and explain why it matters and what it does not promise. The aim is clarity. You will leave with a concrete understanding of physiology, realistic strategies you can apply, and the appropriate expectations about what natural approaches can achieve compared with medication. A recent line of research from Stanford described a small peptide named BRP, which stands for BRIMP2-related peptide. BRP is a 12 amino acid fragment that derives from a larger precursor protein, and in animal models it produced an impressively rapid reduction in food intake, on the order of roughly 50% within a single hour following administration. In those same models, BRP also contributed to measurable reductions in body fat and to improvements in glucose tolerance. What distinguishes BRP from drugs like semaglutide is where and how it appears to act. Semaglutide and related GLP-1 receptor agonists produce widespread receptor activation across multiple tissues. They affect the gut, the pancreas, and the central nervous system, and they alter gastric motility, insulin secretion, and central satiety pathways. By contrast, BRP appears to act primarily at the level of the hypothalamus, the brain's appetite regulation center. Instead of broadly amplifying a gut-derived hormone, BRP may engage central appetite circuits more directly. This difference is consequential. Many of the side effects associated with GLP-1 drugs, notably nausea, transient vomiting, and slowed gastric emptying, are linked to their systemic activity. A compound that targets central appetite circuits with greater specificity could, in theory, produce appetite suppression with fewer peripheral side effects. It could also reveal new molecular pathways for appetite control that are independent of the classical GLP-1 axis. However, it is essential to emphasize that this research remains at an early stage. Most of the evidence for BRP comes from animal studies. There are substantial biological differences between species in appetite regulation and peptide signaling. The efficacy, safety, dosing, and pharmacokinetics of BRP in humans remain unknown. In short, BRP is a promising scientific lead, not a ready-made therapeutic option. To contextualize why BRP and other developments matter, you need a practical understanding of GLP-1 itself. GLP-1, or glucagon-like peptide 1, is a hormone released from L cells in the distal intestine in response to nutrient ingestion. Its physiological roles are coordinated and complementary. First, GLP-1 enhances glucose-dependent insulin secretion. When blood glucose rises after a meal, GLP-1 amplifies the pancreas's release of insulin, which helps maintain glucose homeostasis. This property explains part of why GLP-1-based medications improve glycemic control in type 2 diabetes. Second, GLP-1 activates satiety signals in the brain. Through neural and humoral pathways, it informs central appetite centers that nutrients have arrived, reducing hunger and helping to terminate eating episodes. This central effect is a primary reason GLP-1 receptor agonists reduce food intake. Third, GLP-1 slows gastric emptying. By delaying the transit of food from stomach to small intestine, it prolongs the sensation of fullness after a meal. This mechanical effect contributes to reduced caloric intake over time. A crucial limitation of endogenous GLP-1 is its brevity. The natural hormone is rapidly degraded by the enzyme dipeptidyl peptidase 4, DPP4. Native GLP-1 circulates for only a few minutes before it is cleaved and inactivated. That means your body produces GLP-1 in brief pulses, typically in response to meal ingestion, rather than maintaining a sustained high-level signal. Pharmacological agents, such as semaglutide, are engineered to resist DPP-4 degradation and to have prolonged half-lives. They can produce continuous receptor activation over days or longer. That steady, elevated signaling is what produces the large and durable effects on appetite, weight, and glycemic control, but it also explains the broader physiological footprint and the side effects of these medications. If the goal is to enhance GLP-1 activity without drugs, the objective is not to replicate pharmacological continuous receptor activation. Instead, you can amplify the body's natural pulses and improve the conditions under which GLP-1 is released and acts. In practical terms, that means optimizing meal composition, the gut microbiome, lifestyle behaviors, and exercise patterns. The following strategies are evidence-based and actionable. Dietary protein is one of the most reliable stimuli of GLP-1 release. Amino acids stimulate L cells through nutrient-sensing mechanisms in the gut. Specific amino acids, such as glutamine and phenylalanine, are particularly effective at triggering GLP-1 secretion. What this means for your meals is straightforward. Include a meaningful source of high-quality protein at every eating occasion. That can be animal protein, lean poultry, fish, eggs, dairy, or plant-based options, such as lentils, chickpeas, tofu, and tempeh. A reasonable, practical target is 20 to 40 grams of protein per meal for most adults, adjusted for body size and goals. The satiety benefits are twofold. Protein directly stimulates GLP-1 and other satiety peptides, and it increases thermogenesis and lean mass retention, which supports long-term metabolic health. Fermentable fiber is a critical determinant of GLP-1 signaling because of the interaction between dietary fiber and the gut microbiota. Beneficial gut bacteria ferment resistant starches and soluble fibers into short-chain fatty acids, SCFAs, such as butyrate and propionate. These metabolates act on L cells to stimulate GLP-1 release and also serve as signaling molecules that influence appetite and metabolic function. Practical dietary sources include oats, legumes, beans, and lentils, and a broad array of vegetables such as broccoli, Brussels sprouts, and onions. Whole grains and minimally processed plant foods are particularly useful. Aim for a daily fiber intake in line with public health recommendations, typically at least 25 to 30 grams per day for many adults, with an emphasis on fermentable soluble fibers. If your habitual diet is low in fiber, increase intake gradually to reduce gastrointestinal discomfort while allowing the microbiome to adapt. Fats influence GLP-1 indirectly by slowing gastric emptying and extending the duration of nutrient exposure in the gut, which can enhance hormone release. Monounsaturated fats and long-chain polyunsaturated fats tend to be preferable in this context. Sources include olive oil, avocado, and fatty fish rich in omega-3 fatty acids. The practical approach is not to consume excessive fat, but to include moderate, high-quality fat in meals that already contain protein and fiber. Combining these macronutrients creates a more sustained and effective satiety signal than any single nutrient consumed in isolation. The most reliable way to maximize endogenous GLP-1 pulses is to combine protein, fermentable fiber, and healthy fats in the same meal. For example, a meal of baked salmon, a lentil salad with vegetables and olive oil, or yogurt with oats, berries, and nuts will produce a more prolonged satiety signal than a meal based on refined carbohydrates alone. Physical activity increases GLP-1 levels in the short term and may improve GLP-1 receptor sensitivity over the long term. Both aerobic exercise and resistance training can be beneficial. From a practical standpoint, incorporate regular exercise into the weekly routine. Aim for at least 150 minutes of moderate-intensity aerobic activity per week, plus 2 or more sessions of resistance training, adjusted to individual ability and health status. Exercise complements dietary strategies by improving insulin sensitivity, promoting lean mass, and enhancing the hormonal milieu that supports appetite regulation. Sleep deprivation and chronic stress negatively influence appetite-regulating hormones and can blunt GLP-1 responses. Inadequate sleep increases hunger and cravings and impairs insulin sensitivity. Chronic stress elevates cortisol and can dysregulate eating behavior. Prioritize consistent, sufficient sleep and use evidence-based stress reduction strategies, such as mindfulness, structured physical activity, and social support, to support hormonal balance and appetite regulation. It is important to set realistic expectations. The strategies described, optimizing protein, adding fermentable fiber, including healthy fats, exercising regularly, and managing sleep and stress, can enhance your natural GLP-1 signaling and improve satiety and metabolic health. These approaches operate within the body's physiological constraints, promoting moderate, sustainable changes that are unlikely to produce dramatic, drug-level effects on weight in the short term. Pharmacological agents like semaglutide are designed to resist enzymatic breakdown, achieve sustained systemic levels, and produce continuous receptor activation. Their weight loss and glycemic effects are substantially larger than what natural strategies typically achieve. That is not a failure of natural approaches. Rather, it reflects the fundamental biochemical differences between endogenous pulsatile hormone secretion and engineered long-acting agonists. Emerging research, such as the BRP findings, suggests that new, more targeted approaches may be possible in the future. Approaches that could combine strong appetite suppression with fewer peripheral side effects. For now, however, those developments remain experimental. In practical clinical or personal decision-making, the choice between lifestyle strategies and pharmacotherapy should be individualized. For some people, natural strategies are sufficient to achieve meaningful health improvements. For others, particularly those with severe obesity or poorly controlled diabetes, medication will often be the most effective and appropriate option when used under medical supervision. The optimal path can also be blended. Lifestyle modification plus medication frequently yields the best outcomes in terms of weight loss, metabolic improvement, and long-term health. GLP-1 drugs provide continuous, high-level receptor signaling and can drive substantial weight loss and glycemic improvement. They are engineered to resist degradation and act systemically. Your body's GLP-1 system produces short, tightly controlled pulses in response to meals and other stimuli. These pulses can be amplified by meal composition, fiber-driven microbiome activity, healthy fats, exercise, and sleep. Research discoveries such as BRP may point toward targeted central nervous system interventions that could change future treatment options, but translation from animal models to human therapy is not guaranteed. Most importantly, you are not powerless in this system. You influence GLP-1 signaling every day through what you eat, how you move, and how you manage sleep and stress. Those influences operate within physiology, so their effects tend to be more moderate and better regulated than pharmacological interventions, but they are practical, sustainable, and foundational to long-term health. Are you using your biology to control your appetite? Or are you relying on willpower alone and hoping it works? Those are two very different approaches. If this material clarified the distinction, consider engaging with the content to help it reach others who need a clear, practical explanation of how GLP-1 and appetite regulation work. Like this video if you found it helpful. Subscribe for regular, evidence-grounded breakdowns of health science and practical strategies you can actually use. And if you want a deeper dive, watch the next video on the screen, where the mechanics of insulin resistance are explained in detail, and the most effective steps for beginning to reverse it are laid out. If you're thinking about medications such as Ozempic or Wegovy, speak to a licensed healthcare provider who can evaluate risks and benefits in the context of your medical history. New research will continue to refine our understanding, and as it does, the combination of sound science, sensible lifestyle habits, and appropriate medical care will remain the best path to sustained metabolic health.